Hello, everybody. My name is Veneta Callpani. I'm majoring in biology, brain science. Today, I'm going to talk about vaccines and COVID-19 pandemic. Today's agenda is what is a vaccine, the common virus vaccines, what is an mRNA vaccine, and then specifically, I'm going to talk about Moderna, Pfizer, and Johnson & Johnson vaccines. So what is a vaccine? Vaccine is a substance that helps the body to activate the immune system to help fight against the disease. It can elicit innate and adaptive immunity system. Now, the innate immunity system is the first line of defense. It is non-specific and it cannot induce immune memory. However, it can signal and activate the adaptive immunity system, which indeed it is very specific, and it can induce immune memory. The adaptive immunity system consists of humoral response, which is antibody response mediated by B cells, and also the cellular response, which is actually happens within the infected cell and is mediated by T lymphocytes. Before the vaccines, smallpox was an infectious disease caused by viral virus and had a very high mortality rate. At that time, violation was practiced to help eradicate the disease. This was done by taking a small scab of the disease and, inoculated it into a non-infected patient. This was very dangerous because the patient could die from the procedure and the mild form of the disease could actually be spread and cause an epidemic. So when did the idea of vaccines come to life? The idea of vaccine was introduced in 1796 by a British physician named Edward Jenner. What he observed was that the milkmaids that were infected by the cowpox virus, they were actually from the smallpox virus. So since the cowpox virus would cause only a mild disease in humans, he used that virus to vaccinate those that were infected by smallpox. And vaccination can actually help eradicate viruses. And you can see here in case of measles, for example, the introduction of vaccination in 1968 made the virus, like the cases to decrease until it was eradicated around 1998. And we have, like other examples such as polio or influenza type B. So what are the common virus vaccines? One of them is attenuated vaccines, which contain small amount of the weakened virus that triggers the immune system into fighting it off without actually causing sickness. This vaccine is often used to prevent viruses like chickenpox, smallpox, and measles. Protein subunit vaccines contains harmless pieces of the virus proteins. They don't have any live component of the viral particle, and this method is used when creating hepatitis B vaccines. Another type is inactivated virus vaccine, which actually is being used for the COVID-19 virus. Uh, is the Sinovac vaccine, which contains the kill COVID-19 virus. So basically the virus, the nucleic acid of the virus is destroyed, so the virus cannot replicate. However, the antigens are being intact, so like the spike protein, so our immune system can recognize it and start building immunity against it. A disadvantage of this is that it requires a booster shot to maintain immunity. Viral vector vaccine was introduced in 1972 when Jackson et al. Created recombinant DNA from SV40 virus by genetic engineering. A modified version of the virus is used as a vector to deliver genetic code for the antigen. The major vectors used are retrovirus, vaccinovirus, adenovirus, coronavirus. Examples of these vaccines are Johnson & Johnson vaccine and AstraZeneca vaccine, which I'm going to talk about it later. A disadvantage of this vaccine is that the host might have immunity to the viral vector due to the prior exposure. And then we have mRNA vaccine, which introduces an mRNA sequence that codes for a specific antigen. Now, what is mRNA and what are the advantages of using mRNA? We utilize mRNA because our body uses this to actually form proteins. And this is called the central dogma. So as you see here, but also here in the cell, the DNA in the nucleus is being transcribed into mRNA, and then mRNA is being translated, and it goes through all these processes to form proteins. The advantages of using mRNA is that, first of all, it is non-infecting and non-integrating, which means it cannot integrate into our genome. Also, it has the potential to be rapidly manufactured. It can be controlled, and also the mRNA can be degraded by just normal cellular processes. So if it has all these advantages, why hasn't been used before? Well, because there are many challenges that scientists had to overcome, and that would need all this advanced and sophisticated technology, because they have to come up with a way to add the 5 capping and the 3 poly A tail into mRNA, because both of these help to keep the mRNA stable, but also ensures for efficient translation, protects the mRNA from degradation, and prevents activation of antiviral immune defenses. And besides these, they also had to come up with the right building of the lipid nanoparticle that basically protects the mRNA, stabilizes it, but also helps the mRNA to get into the cell, so to enter the cell. So the mRNA has not been used before. However, it has been tested before. And mRNA was studied before for various different viruses such as influenza, rabies, Zika, and cytomegalovirus. As you can see here, in the time in 2016, when there were many cases of Zika virus, researchers started to do some preclinical and clinical trials for some vaccine candidates, and you can see here that mRNA was one of them. And it was shown to be very promising and successful, especially after they made the nucleoside modification and after they added this lipid nanoparticle. However, there were setbacks because of the lack of funding. And here we come to Pfizer and Moderna vaccines that are widely being used in the United States. So here we have the mRNA vaccine with that encodes the version of the spike protein with 2 proline modification. And this is the lipid nanoparticle that I was talking about that, like codes the mRNA, and it stabilizes, it protects, and it protects from degradation, and it also helps the mRNA to enter the cell. Once the mRNA is in the cell, it will go through translation in the ribosome, and it will form these spike proteins, which once the spike proteins are outside of the cell, they're gonna induce both the humoral and the cellular immune response. Both have shown to be 91.5% to have 91.5% efficacy in preventing COVID-19 illness, and they both require 2 shots. However, Moderna is less sensitive to temperature, therefore, it's easier to store and to distribute. And they were shown some cases with allergic reaction to both the vaccines. However, those cases were rare. And then we come to the viral vector vaccines, such as Johnson & Johnson vaccine and AstraZeneca vaccine. They use adenovirus viral vector to introduce the spike protein sequence that codes for the spike protein. And to overcome the challenge that we have immunity for adenovirus viral vector, the researchers use the ADV6 adenovirus serotype. So again, once in the cell, it's going to be translated, and it's going to form the spike protein, which then is going to induce the immune response, both humoral and cellular. Because it's using DNA, DNA is more stable, therefore Johnson & Johnson vaccine is more stable. It does not require ultra-cold storage. It has been shown to be 85% effective in preventing hospitalization and death, and it only requires one shot. Now, there have been cases reported that people were developing these blood clots. And even though it was rare, it was very interesting because these patients had very low count of platelets, which means actually they would have trouble forming blood clots. And this was, like it's considered like a rare phenomena. And scientists and researchers they have some hypotheses. However, like they're still looking into this. So far, however, we know that the risk is less than 1 in a million. What are the relative risks and the side effects getting the vaccine versus COVID-19? So I hope by now with all this information you can understand that getting the vaccine, it doesn't mean that you're getting infected with the virus. Therefore, the side effects that you might experience are short-term, or like headache or fatigue or muscle ache, or like swelling in the site of injections, versus if you get COVID-19, you're actually getting infected, and this is affecting your organs, and it's like more long-term. And of course, like the possibility and the extreme case would be death. So you might die from COVID-19. And now we are at the end, and I really hope that this information and all this research will help you make an educated decision to get the vaccine. I want to thank my group for their contribution. And of course, I want to thank my professor, Ethan, for all the feedback and help. And thank you all for the participation. And these are my references. So thank you.